What I'd like to do today is update everybody on our Luminate programs in diabetic macular edema. When we look at DME, anti-VEGF is certainly the standard of care, but when, as many people have mentioned as well today, anti-VEGFs work well in about 50% of the patient population with diabetic macular edema. The other 50% of the patients don't respond adequately to treatment, and it's very interesting that how the patients respond in the first 3 or 4 months of treatment is a strong predictor of how they're going to do 1 year, 2 years, and even 3 years out. So a poor response up front tends to be a poor response even out to 3 years of follow-up with continued anti-VEGF. Why is this? Because DME is a complicated disease. Some patients are mostly VEGF-driven, other patients are mostly inflammation-driven, and other patients still are mostly neurodegeneration-driven. Clinically, they all look the same, but they don't behave the same to treatment. What underlies all of these processes in DME is oxidative stress, which you've heard again and again today on several presentations. Oxidative stress drives the DME process, and this is 4 pathways in oxidative stress: vascular leakage, neovascularization, inflammation, and neurodegeneration. Anti-VEGFs certainly cover the first 2 very well. Steroids with all their complications tend to cover number 3 very well. Nothing covers neuroprotection, and nothing covers all 4 pathways at the same time. What's interesting is the way Luminate works, or Risuteganib works, is up at the oxidative stress switch. It actually mitigates the oxidative stress switch so you can actually control all 4 pathways at the same time. To be able to control all 4 pathways gives you options you don't have now, and this has been worked out very carefully over the last several years with 5 different university collaborations. Integrins are very important in not only in ophthalmology, but throughout medicine. There are about 6 anti-integrin drugs on the market overall. They've been described for about 30 years, and RGD class anti-integrin drugs in particular are very important for the oxidative stress switch, as you'll see in a moment. When you look at these pathways, if we can mitigate the oxidative stress switch, if we can turn this switch off, you should be able to control all 4 pathways underlying oxidative stress. If you look at the angiogenic pathways first, this is data that we've shown before. In pre-retinal angiogenic models, the drug is about 4 times more anti-angiogenic than Eylea is. This you've seen before. These next pathways you probably have not seen. If you look at inflammatory pathways, the drug gives you about an 80% downregulation of the complement 3 receptor directly. The complement 3 receptor is also known as alpha-M beta-2 integrin. And from genomic analysis between Campochiara's group at Hopkins and Julie Cornfield's lab at Caltech, they're able to demonstrate this. Now, the biggest one is neuroprotection. You say, well, if you can block the oxidative stress switch, you should be able to abort apoptosis and provide some neuroprotection. A lot of work has gone into this over the last several years between 5 different labs. One of the first labs that showed this was Hugo Quiros' lab. Basically, what they showed was if you take 3 different layers of the retina, neuroretina, the Mueller cells, and the RPE cells, and you expose these tissues to different kinds of neurotoxins, pretreatment with Luminate is protective to the tissue. This work was also done again in a different model by Glenn Jaffe's group at Duke, and it showed the corroborating evidence again that in Glenn Jaffe's group, it's not on this slide because he hasn't published it yet, showed very similar evidence that the drug was neuroprotective against cigarette smoke toxin as well, whether it's in mouse tissue or human tissue. Barry Cooperman and Chris Kenny's lab at UCI went a step further, and they said, okay, if this drug is neuroprotective, how is it behaving on this tissue? When you looked at the Mueller cells, what they found is treatment with Luminate increased the viability and growth of the Mueller cells, decreased the reactive oxygen species in the Mueller cells, and actually increased the viability of the mitochondria, increased the power source of the cell. The group at Caltech also has shown that they're turning on the repair mechanisms via the MAF pathway. That's all fine and dandy, but let me show you what's happened also with the clinical pathway going forward. If you can mitigate all 4 pathways at the same time, what does it mean for our lead candidate, Luminate for diabetic macular edema? To date, we've done 2 phase 2 studies in 216 patients. Risuteganib, or Luminate, has been equivalent to anti-VEGF in both studies, with half the number of injections, 12-week durability, and the interesting thing is the patients who don't respond well to prior anti-VEGF treatment are the ones that get the best response to Luminate, because you're covering pathways you don't cover now. If you look at the two arms that are going into phase 3s by the first quarter of next year for DME, Avastin versus 1 milligram luminate, you see for all comers, the visual acuity is about the same. Avastin works quite well in the treatment-naive patients, but in the patients who've done poorly with anti-VEGF before, those are the patients where switching mechanisms of action and covering more pathways gives you a further benefit. And these patients on average had 12 prior anti-VEGF injections and 20/60 to 20/70 visual acuity. Those are the patients that give you the best visual acuity response with Luminate. We had our end of phase 2 meeting with FDA about 3 or 4 months ago. The study design for phase 3 is very similar to what we've already done in phase 2s. Same population, same primary endpoint, same secondary endpoint. So we're going to basically be doing a non-inferiority study for all comers for the primary, superiority in the non-responders to anti-VEGF, 12-week treatment schedules because it's worked in both phase 2 studies already. In summary, you have a broad spectrum treatment. It works very far upstream in the process, so you can affect all 4 pathways, including neuroprotection. With this, you have the ability to treat the anti-VEGF non-responders in DME. You get 12-week dosing in both phase 2 studies, and you have a very clean safety profile so far with close to 1,200 human intravitreal injections and close to 400 patients to date. Thank you.